Yes.
Yeah, we're all good now.
Thank you very much for coming.
It's no problem. No problem.
I'm going to make you co-host.
Is this just running on a normal meeting link, or is it on a webinar type thing?
Yeah, exactly.
Okay.
Just a quick question before we start. How should I call your company? Is it for the biomaterials or for the medicine limited?
We trade as 4D Biomaterials.
For the biomaterials, right?
Yeah, it's we trade. Yeah, that's the we officially it's 4D Medicine trading as 4D Biomaterials, but that's a bit of a mouthful. So 4D Biomaterials. That's how I branded it up on my slides anyway. So
perfect.
Shall
we try sharing your slides just to see if it works?
Yeah, just give me I'm just moving things around screens.
Sure. No problem, no problem.
Should be there.
Yep, it's there. That's perfect.
Oh, I see that. So you can see
I can see your pointer. Yep, perfect.
Cool. Just want me to leave that up? Is that the easiest thing to do?
Yeah, I would think so.
Okay, it's just my camera's over there, and my zoom's over here now, so it just.
Oh right, there will be. Okay, that's not a problem.
Yeah, it's just it's the easiest way to set up my system so that when I'm actually talking about my slides, I'm looking at the camera. But otherwise, you end up with this. You know, if I do that, then you can, you know, kind of you get that. It's a bit of a mess, but I can put everyone in the corner there. But
I guess if there are all these so many Zoom meetings, we are now all used to people looking at different places than the camera is and so on. So.
Oh yeah, it's you get used to this, right?
Yeah. Okay, I'm just going to tell Yusuf with us if he can let people.
What's the audience?
We got 200 signups.
Okay.
We'll see how many people do turn up. Normally the ratio is around 40 to 60, sometimes 70%. We send reminders 2 days before the event and a few hours before the event, so people get the link and so on.
Do you know what you know? Is it a technical audience or an investment audience, or what sort of people are they likely to be?
Basically, I think it's mainly.
But this corner of the garage will be on.
All right. Welcome, everybody. I think we are going to get started almost now. It's 3 minutes past 6 o'clock. So welcome, everybody, to this Innovation Forum event, Biomaterials at the Interface with the Human Body. The Innovation Forum is an accelerator for healthcare ventures. We also organize events like the one you are attending today. Innovation Forum is supported by a global grassroots network of over 10,000 innovators who seek to improve human health and well-being by translating cutting-edge science into innovative products and services. We focus on the future of the evolutions of today's technologies, which range from the nascent stage to the cusp of commercial application. In that regard, today I'm delighted to be joined by Professor Andrew Dove, Professor of Chemistry at the University of Birmingham and CSO and co-founder of 4D Biomaterials. Professor Andrew Dove is a world expert in biodegradable materials and polymers at the School of Chemistry in the University of Birmingham. The creation of 4D Biomaterials was the culmination of over 12 years of research, and now Professor Dove is determined to see his novel materials technology translate into commercial success, delivering better treatment experiences and outcomes for medical patients across the globe. Professor Dove is delighted to have you in our event today, and we are all waiting to hear your presentation. The virtual floor is yours.
Thank you very much. Thanks for the invitation to talk to you about this story, and I'm happy to answer your questions later as well. My plan is to try and talk you through the journey that we went through, so the story of how we went from just doing a bit of academic research with a vision of where we might go to, but not necessarily what it's ended up as, to where we've got to. I call it from lab to clinic. We've not quite got to the clinic, but that's certainly the aim, is to take this technology through. So as Andrea said, I'm at the University of Birmingham, where I'm Professor of Sustainable Polymer Chemistry, and I'm also a part-time Chief Scientific Officer of 4D Biomaterials. But I should point out that this work actually, I was at the University of Warwick for the first 12 years of my career, and we started a lot of this work there. So I'll talk about at the start with at Warwick, but we transferred it all over to Birmingham around the time that we spun out the company, and I wouldn't recommend to anyone to spin out a company whilst in the middle of moving universities, but that's a slight complication of this story. Hopefully you can see the top corner of my screen as well, as you might miss just a few slides, slide titles, but there's nothing to worry about. My academic research is really summarized on this slide. So typically we look at two different aspects of how we can make sustainable polymers and recycle plastics from sustainably sourced monomers, efficient, non-toxic chemistries, and how we can degrade polymers. And then how we can make them into interesting materials. And as part of this mission, really, we were really focused when I started on on this degradation aspect, and really what links pretty much all of our research together is this degradation aspect. And the stereochemistry I can skip over because it's very much my academic side of things. But when I started in 2005, people just weren't really interested in how you could make plastics degrade from either current petrochemical plastics degrade, and the reality of taking a sustainably sourced monomer and creating it into a commodity plastic was just it was a million miles away. I just didn't feel we were going to do it. So we needed a different focus for our research where we're going to create these polymers that degraded. And it really came in the biomaterials area of the sorts of materials that we make. My theory was, well, if they're made from sustainable sourced things that you would eat or things that you would find in the body, and we make them into a material, when they degrade, they should degrade to things that will be safe to have in the body, and that was what we started off, in our focus on. And I'll take you through some of the accidental things that we found as we went through this research program. It certainly we didn't set out with the idea of creating a spin-out company, let alone in the area of 3D printing scaffolds, but I'll come on to that as I go through the talk. What we did recognize pretty early on is that but the tissues of the body have very different mechanical properties to the most commonly used plastics. So in this little plot here, I've plotted modulus, which is flexibility against strength. And what you see is that, you know, most tissues are actually relatively flexible, but still quite strong. Whereas PLGA, which is the preeminent polymer in tissue engineering, is not very flexible and is really not that strong compared to the things in the body that aren't that flexible. And so we start to think about what would be the ideal, characteristics of, you have no inflammation, there'd be amorphous, these material could surface erode, so you can predictably see what's going on. They would have some elasticity in them, with more elastic than the PLGA. And of course, we need to maintain the non-toxic byproducts as they occur. And if you make this into a little spider diagram and rate out of 0 to 3 for each of them, based on, this is very much a matter of opinion, then PLGA looks a little bit like this. The ideal material is this lovely pentagon. PLGA it's really not an ideal material for tissue engineering replacement. So really, what we set out to do is to try and come up with something that looked a little bit more like this. And I'm going to cut a really long story short. What we're going to work on, and what we decided to work on, were aliphatic polycarbonates, which are just a different type of synthetic polymer we thought could be applied as a biomaterial. When you do the same sorts of analysis on, of where these sorts of different materials might lie in this spider diagram, okay, they're not perfect materials, and I might be biased in my scoring, of course, right? But this is a much more pentagonal shape, much more closer to perfect shape. When we look at the range of materials properties that we can potentially get from these types of materials, then the range is absolutely huge, and so you can cover an awful lot more of those potential tissue types that we might want to look at regenerating. So to us, it struck us as a really obvious place to look at. And there are chemistry reasons why we went down this route as well as the materials reasons. And so for the non-chemists in the room, this is probably a terrifying a slide, but really I won't dwell on it. This is the platform that we developed of having a cyclic carbonate with a functional side chain that you could polymerize to make these oligomeric carbonates. And the point of this is you can take any color of the rainbow pretty much, and you can make anything that you want on this side chain. And what that enables you to do then is to start to create some really complex materials. But what we really got interested in are these photoreactive groups, so things like an allyl group or an alkyne group or an epoxy group on the side chain, because then you can start to do interesting things with them. And what we got interested in were photocurable materials. And so what I put here is, this is, all of you will know what photocurable materials are. So, something like a nail varnish is a photocurable material. You apply it, you'll stick it into a UV chamber, you'll come out with a nice, nicely colored nail. What we're trying to do really is take that away. I'm not sure that's disappeared, but create a material from these carbonates, photocure them into a solid material, and that will enable us to do some interesting things with them. Among which we got led into by collaboration in Warwick, when I was there with James Covington, among which would be being able to photocure them into 3D printing. And so if you start to look at the 3D printing landscape, you start to realize that there's an awful lot of potential to impact in medical and dental. Currently, we're looking, if you take this pie chart of where 3D printing is applied, you've got about 16% of application area into medical and dental. Of that, a vast majority of it is actually in these things of surgical guides, an anatomical model, low-cost prosthetics and dental crowns, with a very, very small amount of that being implants, of which most of the implants are metallic-based, not polymeric. And so it seems to me that there is a huge opportunity here to potentially increase the amount of plastic type, biodegradable plastic medical devices. 3D printing clearly gives you huge advantages in the sorts of shapes and complexity that you can create. And so what we realized was that there was a huge gap in the market. And what typically would be if you had a degradable polymer resin in the literature at the time was you'd make a polymer, a degradable polymer had a reactive end group, and you'd polymerize those reactive end groups to get your network of your polymer. But when you degrade it, only the original degradable part would degrade away. So you'd be left with all these long chain polymers in your body, in the tissues. Now, also a factor of this is it would leave acidic residues. So they've got potential irritants in the tissues that really it's not going to be got rid of by the body. And so it really was clear to us from very early on in this challenge, as soon as we started to recognize there was an opportunity in 3D printing, as soon as we started to look into the materials that were there, that one of the big challenges was the materials design. And so it was clear, and I'm not sure I would have phrased it as such, at a time in probably 2009, 2010, when we first started to really think about this, but there's an unmet need for a fully degradable 3D printable material. And that is relatively still true today, and that's really where our opportunity started to come from, and we started to realize. But I think still at this point, the research was very academic. It was understanding what we could do and how we could do it. And so you start to think about this, all the sorts of different ways that you can 3D print things, and how are we going to make a useful material by 3D printing. And there's lots, the more you look into 3D printing, the more different types of instruments there are. So you can look at these common ones: 2-photon lithography, stereolithography, fused deposition modeling. This is just a sample of the different types, but it's a useful example for me. And you can look at the resolution that you can easily access with these sorts of different printing types. And so FDM, typically it's hard to get resolutions below 100 microns. It's about the nozzle head that you can get, same with direct laser, right? 2-photon, you can get really low resolution. In the middle, you've got stereolithography. Now, what sort of resolution do you need for tissue regeneration? Probably somewhere in this sort of region. This would be overkill. This would probably not be enough. Cost is where the major factor comes in, and where you start to hone in on, actually, if we want a scalable solution, yeah, these are cheap, but they don't give us the resolution we want. These are really expensive. They probably give us too much resolution. You can balance up resolution and cost of these sorts of instruments, and they become really accessible then to start to translate and think about resins. And it uses these pre-polymer type resins that we talked about, that I just briefly introduced, to create to solidify materials from. So it's something that you can then start to go and think about translating and using, because it's in the right resolution area and it's the right cost area. So how these work is incredibly simple. As printers, this is one we actually made in my lab a few years ago now, but really it's just it's a digital light projection projector, like the sorts we'd have if we were all sat in a room together talking about this. You project shapes into a prism, which then redirects them, and sometimes you have lenses in there as well to focus them into a resin tray, which will contain your resin, and then a Z stage which moves up and down. Just to animate this, what you have is your 3D shape that you can slice into 2D layers, and then you take each one and you push it through your light system into the resin. The resin cures into the shape that you're projecting, and then the Z stage moves up to give you a new the shape that you wanted, but inverted. And this is just a quick video to show you it in action. This is inside one of the 3D printers in my lab in Birmingham. That you can see what you're doing is you've seen just under here, you'll see the flashes of light. We'll change the angle in a minute, and you'll get a much better angle on seeing what's going on. But this is the resin bath. You can see in this particular way, it's the resin bath that also moves to as the printer moves up. But here, what you can see, here's the resin, here's the liquid resin, here's the light projecting, and you can see the solid part being moved down. And the student that made this was French, and so of course it had to be the Eiffel Tower that we printed. But still, it's a demonstration of how you go from the liquid to the solid using this 3D CAD model. You split into slices, and then you rebuild out into the material. So as I say, this is a well-known technique. This is an old technique, but the question is, how do you then take a material that you could apply, and how do you take that forward? And so whenever you want to come up with a new ink, as we often call them, for stereolithography, there are lots of things you've got to think about, especially in this area. So we need to have a material that had controllable degradability. We need to have a material that we could obtain mechanical properties, as I introduced at the start. This is probably one of the key aspects of it. You have to actually make it printable, and that's actually, you can't lose track of that. And if you want to make a biomaterial, you actually have to make a material that's biocompatible. Then you can start to add in some advanced properties. Now I'll show you briefly some of the shape memory properties that we started to add in, so you can start to think about things like minimally invasive surgery, where you can take a 3D printed shape, crush it down, and let it respond back up once it hits body temperature. So how do we go about getting good degradability? Well, we want to have surface erosion. So you want a material to erode from the outside in, not the inside out, like you would commonly see with a PLGA. And this comes down to the fact it's cross-linked. It comes down to the fact that it's a polycarbonate, so we can control that. So this video here is just a video of a 3D printed part that you can see just effectively looks like it's dissolving away. This strut over here just getting thinner and thinner and thinner and thinner until it gets so weak it buckles and then breaks and dissolves. And here's some of the data from it. You can see a very linear reduction in that, and here's the mass loss data. So you can see we're getting controlled degradation. The degradation control is dependent on the material that we use as well, so we can take that forward really effectively. So we know these materials are going to degrade, at least in hydrolytic solution, which indicates they should degrade in the body, which we've taken further, and I'll talk about in a few minutes' time. We want materials that we can control the mechanical properties, and again, through controlling that chemistry, you recognize that you can control from something that's very soft and flexible up to something that's actually quite hard and less flexible. So we can go from materials, this material, if you have it in your hands, is very much similar to fatty tissue. This material is much closer to bone in terms of its strength and its modulus. And so you can control anywhere through that, because we can control the chemistry of the resins and the formulations that we use, that we photocure, to take that material into a range of different areas. If you want to print it, you've got to get layer control as well, which is a bit of a trick. So the way this is done is by adding a photoinhibitor, and the photoinhibitor is not to stop the resin curing, it's to stop the light penetrating too much. So if you don't have a photoinhibitor in, what you get is you get this addition where you'll add the shape you want, but when you change shape to cover over this side, the material will cure right through to the top of the stage. So there's some trapped material, so you end up with this elephant-like type of structure. But if you do it with a photoinhibitor, and all this does is it limits the wavelength of the light to not going much past the single layer, then what you do is you pull this out, you will not have the resin trapped in there cured, and you'll get the shape that you desire. So the way we came up with this was a little bit bizarre. This was probably the biggest trick of all in coming up with a biocompatible resin. Most of the things you want to work with, we want to work with a blue light to cure, and therefore you'll need a red dye to attenuate that light going through. And it's amazing how many different obvious dyes are water soluble but not oily. Now, resins are typically oily based on the sorts of materials they are. And it's very, very difficult to come up with something that will actually go in there. And the secret for us very much was this is a true story, that I was out for a curry with some friends. We've been really struggling with this problem, and the dinner came out, and one of my friends had ordered a chicken tikka masala. It was one of these curry houses where there was a really thick layer of oil at the top of it, but the oil was bright red. And that was the eureka moment of, hang on a minute, if that oil's red and you're about to eat it, then this is a red oil soluble dye that is biocompatible. And you can do a little bit of research and you can find out what that red dye is, and it's actually a mixture of these two compounds that are paprika extracts. And then you can do the science on it, and you can prove that you can add some of this paprika extract and it absorbs the light, and you can limit the cure thickness, the penetration depth of your material based on the amount of photoinhibitor that you can put in. So you can now start to control things, and so we can start to create really complicated shapes from our materials. And so here's just a couple of examples of what we can do. And one's just a 3D printed cube that's very difficult. You wouldn't be able to make it by any other than 3D printing types. This is more of a stent type circular mesh that you might want. This is a 5p piece for scale, right? So these things can be really tiny and really complicated to access by any other way. Now, there's no point in making a biomaterial if it turns out it kills cells, and we got really lucky that actually this is quite the opposite. That cells really love to proliferate on our materials, and so we can grow, we can put cells in the presence of these materials, and they'll quite happily proliferate all over the scaffolds. You can see this in these confocal images of reconstructed of the this is meant to be a video, but it doesn't seem to be working, but of the 3D scaffold. Now that was a video that's twirling around, and here's some cells and a lower thing. So the cells really like this material. We expected they would based on our knowledge of these materials already, but it was important to do. And I've always been someone, I'm a chemist by training, a synthetic chemist, and so we've pretty much already gone a long way from my expertise zone. But if you want to translate something, you can't just keep making more materials and expect someone to pick up the gap. There's a huge gap between making a polymer or a plastic in our case, even in 3D printing it, and it being useful. You have to understand its mechanical properties so you can talk to people about it and identify where it might be used. If you want to think of it as a biomaterial, you have to start proving it's biologically compatible, and even this as a demonstration isn't really enough. And so we decided that we'd go further, and I got an animal license so we could do some initial in vivo work. And so we were able to take these materials into a subcutaneous rat model as well. And so we were able to implant them into the backs of rats, and we could then look at how those materials affected healing. And what was amazing to us is that these were some of the best materials we've seen in the literature in terms of the lack of inflammatory response. So there's almost no fibrous capsule around the implant. We already see adipocytes infiltrating the scaffold. So when we print them in 3D, we get fatty cells going right throughout the material, and really get great healing in terms of these materials. So we want to take it really a long way before we could think about actually trying to translate it. I think it's one of the pieces of advice I got when we were starting to think this might be really interesting, it might be something to do, was that you need to prove as much of it as you can and de-risk as much of it as possible before you try and translate it. Otherwise you leave yourself with too much to do when you start to bring in the investment money and expecting to make it into a commercial product. But the other thing we wanted to do with it was as I already mentioned, is make it so it's shape memory, so you can get it to respond. And this is a bit of science about what shape memory is, but basically you can take a material, you can heat it and load it, so you can deform it, and then when you cool it, you should fix a new shape in place, and then you unload it, and that shape remains until you heat it when it springs back. So here's an example of a coil that's been made, and then you straighten it out, and then as you heat it, you'll go back through and you'll recover your shape. So we can do that with our materials as well. And so here's just an example of taking a flat piece of material. If we coil it, in this case, you have to cool it down to fix the shape. So you freeze the shape in by going through the transition temperature of that response. So you now got a fixed shape. If you heat it back up, then back up to room temperature, you can see that you get the material respond back to it. So well and good doing it in 2D. Can you 3D print it? Well, yes, you can. So here's a video of a little 3D printed stent type model, which you can take and you can get to respond with just a little bit of heat on it in this case. So you can fold it up, so it's it's tiny compared to its original diameter. Then you get a full recovery back to it with these 3D printed models as well. So it's translatable through into 3D printing. And so, that's the academic research really. And, I'd always wanted to do something that was going to make a difference, or that had a chance of making a difference, rather than going around the same loop and making a new material and saying that that would make a difference. But you get to a point, I think, in academic research where it's very difficult to take it further. You start to ask the question of, of where you're at. So, we've got this, we've got this, we've got this, we've got this, we've got some cool advanced properties. But what's the next step? How do you make it into something that's useful? And this is where you have to get really out of your academic comfort zone and start to do things that are really different. And you have to ask yourself some questions, right? So it's all well and good us making it on a 10 gram, even 100 gram scale, if you're going to sell it, you need to make more. So the question, how on earth are we going to make more of it? How are we going to make it work? And still got to validate how to get into a body. But moreover, does anybody even care? Is this a good idea or not? Could it be, is the market receptive to this? We've identified there's this problem, but does anybody else think there's this opportunity or problem out there? And so what we did, I had a really talented postdoc who's the co-founder of the company. At the time, he's left to pursue an academic career, but he's still involved from time to time. Where he went on the iCure journey, so innovation to commercialization of university research, and that really was transformational for us in terms of testing the idea. So he got paid for 3 months to travel the world when we could see each other and talk to people in person. And he got to ask lots of questions about lots of people about what we got and what they thought was important and what they would be interested in. And this led to this, this great quote from the global director of sales of a leading 3D print company. This material system is exactly what we're looking for to move into implantable devices. It starts to tell you that there's a market out there for the sorts of things that we were thinking about. We've always struggled with whether the business opportunity was as a resin manufacturer, a medical device manufacturer, or a medical device company. And it's still something I think we're still working on as we keep taking this forward. But from here, I'm going to jump a step, because then the next slide really is what the next step of it was. But you start to take further validation and further market research on, which was funded by Innovate UK as a follow-up to the iCure grant initially. But we get great quotes out of this, like, I've been trying to find these sorts of materials, there's nothing out there. This could be the holy grail of a serial entrepreneur that we'd approach as part of this process. And you start to realize that maybe someone does care about it. It then becomes a matter of how on earth do you make this a reality? And it's a huge deal going from an academic lab, even just to take a material from Warwick to Birmingham and repeat it was a challenge. And that's 2 well-equipped world-class labs. How do you take it to an industry lab where perhaps you haven't got the same level of equipment and stuff? And I think a key part of that there is the team, and recognizing pretty early that I was not going to be the person that could realistically translate this and make a business out of it. And I was very lucky, very fortunate to have an old friend that I knew that I'd met in a previous life at Warwick, Phil Smith, who was at a loose end because he'd just left his previous spin-out company. He's a veteran of 3 or 4 spin-out companies, as well as a lot of other business experience. And I said to Phil, hey, what are you doing? How do you fancy doing this? And Phil looked at the business opportunity, looked at the data we got so far, and was interested and wanted to find out more. And so he came on board as our CEO. And that really enabled us to start to think about making this into a business. I don't think, however, the bandwidth to do it myself. I don't have the business expertise to do it. Sitting watching Phil operate is very educational for me in terms of learning how the business world works and the different way of thinking from academia of what we do because we want to do, because we're interested in it, through to actually thinking about how do we make this into a product? What are the key steps along the critical path? What is good enough versus what do we need? Or what do we want? And he really challenges the science and the engineering on that level, even though he's not a chemist or an expert. We're really lucky that we've got, we brought consultancy on board as a way to bring high-level people into the company without them costing the earth. People with a lot of experience tend to demand quite big salaries, and so you can manage that by doing that through some consultancy. It may not be as efficient, in terms of cost for the time that they have, but the value that we're getting out of this approach. This is just one example with Phil Reeves, who's a world-leading 3D printing advisor. We have a part-time CFO, but the real step was, how do you start to make these materials? And it starts to answer this question of how do we scale up? And you need people on the ground in there, and I recognize that I can't manage those people necessarily, partly because we've made the decision to move to Nottingham as a result of some of the COVID challenges that we faced for the lab to set it up. But we got really lucky with a really excellent, experienced spin-out guy, Andy Naylor, who heads up our lab, and Dr. Haley Ang, who's a very experienced chemist. And we've recently hired someone to help out with the administrative side of things. to take this forward. So you build your team and you start to, I do officially, I do 10% of my time for the company. But realistically, these guys are the ones that are making the dream real, and I probably am becoming less involved as we go forward. But I think that's kind of how it needs to be for the products to move. So the other thing that was really important was getting somewhere to do this sort of thing. So, getting distance from the academic lab, I think, is important, and the fact we did it really early, I think, was really useful for us, because the thing I've seen of translating academic research into industry research, it's not just with this project, it's with other projects as well, is that we're really spoiled in academia. We have amazing facilities. We have multiples of millions of pounds of analytical equipment on our doorstep to just go and use and test questions. And when you start to take things out and repeat them in a different lab, everything's just slightly different. It never quite works the same way because something's different. The person's different for a start, the lab's different, the facilities are different. And so things don't always go the way you're expecting them to go, and there's teething troubles with that translational process. And so I think it was, in retrospect, whilst it was probably tougher to do, it's been a really stronger learning experience for us as a company to be able to start early on working in a laboratory that's more than big enough for our current needs, to really start to translate these resins and start to scale them up and address some of the challenges just of repeating our academic research in a different lab, a different place, different people with less stringent conditions, less great equipment, although we're very well equipped, as you'll see from this slide, for a small company. And then start to address some of the problems that perhaps you don't think about as what's good enough for an academic lab is that you can make something, right? The yield's not so important, the cost's not so important. That becomes more important when you're thinking from a commercial angle. And so we're quite fortunate that we're working in an area where the potential profit margin is quite high, and so the cost of our production isn't quite as important as it would be in a different area. But those sorts of things start to really have to wear on your mind as a commercial entity. And I think if you're thinking about taking something commercial early, then you might want to be thinking about some of those things, embedding them into your academic research and not overlooking them quite so much as you might be tempted to when you're just chasing the paper. But I mean, this story wasn't an intention necessarily to spin a company until we realized there was an opportunity to go for. And so, where do we go from here? Well, now is the point where we're at. We have resins that we can make, and we can sell, and we've closed our first deal on selling them. So we're actively selling these resins now. We're exploring a load of other collaborative opportunities through a few different types of business models. But we have now what we believe to be the world's first 3D printing resins that are suited to tissue engineering applications. This is a picture of the resin and a few parts that have just been printed out just to show what can be done. But how do we go from here now? Well, we have to now start to find buyers, more buyers for it, co-develop the materials with them, co-develop devices, and really start to deliver on the business model that we're proposing to work with. So that's all I've prepared in terms of slides to talk through, and I'm happy to take any sorts of questions on. I've just there's a lot of people who contributed to a lot of this through collaboration and co-works both at Warwick and Birmingham. Initially, this was all academically funded, but as we translated, we went through IQ, Innovate, and we've been very fortunate to have some very good investors on our side and following up with us. And of course, this team in 4D that we really, are really critical now to delivering on this. So thanks for your time and your attention. If you've got any questions, I'll happily take them.
Thank you very much. It was an excellent presentation. I love how you've driven the conversation from how everything started in the academic lab to all the eureka moments with the color, the oil, the resin, and then all these very important points on how entrepreneurs should be doing, going out of their comfort zone, especially when it comes to realizing, right, maybe it's not me who is like say, the father or the mother of the idea who is going to bring this definitely forward to its full extent. So that was very interesting, and I would like to ask you a few things about that. But for now, I would like to remind the audience that we are happy to take questions, so you can just post them in the chat and I will read them out loud for Professor Dove. We have one, so I'll start with this one. It says, hello, I would like to ask what we should do for certification. I understand this sort of like FDA, AMI, or UK certification for health and so on.
Yeah, so it's a long process, unfortunately, and we're at the start of that process. So if you want to get, so you can't have a material certified for, approved for human use. Every, a material is approved as part of a device. There are materials lists of, that you can get that have got a prior track record. You can have a materials master file on file with places like the FDA once you've developed a material, so that you can provide the properties and the preclinical data on previous materials, that device and made up material. But really, that is the the biggest battle of trying to commercialize a new biomaterial, is to go through the approval process. And that's part of the reason why we tried to get our material tested not just in vitro but also in vivo, in terms of part of that risk management. Because what you've got to persuade investors to do is part with real money, right? This is not like persuading a grant institution to give you some money. It's not like persuading your peers that this is a good idea and they should give someone else's money to you. This is their money, right? That they are investing either for themselves or on behalf of someone who wants to see a return on that money, right? It's a very different game. And so you've got to take it to a point where you've de-risked it to a point where someone might take a punt on you, right, and give it a chance. But then, they have to understand that there's no guarantees in biomedical materials research, and there is risk in it, but hopefully the rewards are relevant to that, are balanced against that risk as well. So, the reality is, what we are doing is we are working on materials, and we're working on devices. We're working on that ourselves, and we're working on it in collaboration with other people. And we are trying to get those to a point where we're ready to go to preclinical data, and that probably will be within the next year, that we'll be looking for a preclinical animal study. And once we have preclinical animal study data, we'll be looking to go into a human trial with one of the devices, as early as possible to demonstrate the safety and efficacy of it, and then go through that regulatory process hand in hand with the FDA or the MHRA. As we do that, we expect that once we can get one device approved, the second, third, fourth, fifth will be a lot easier, because there is a demonstration that actually this material is all right. All the indicators so far is actually good, not just all right, it's better than the competitors. But it's a very conservative field, and I think you can understand why, because you're putting materials into people, and they can have bad effects if it goes wrong. So it's a conservative field that can be hard to break into, but we're very pleasantly surprised by the enthusiasm that there is out there for this. And this is amongst surgeons, it's amongst biomedical device companies as well as 3D printing companies. So there seems to be a lot of demand for what we've got.
Sounds very exciting. We have another question here. It says, how do you think your resin will work in the central nervous system? For example, scaffolds for a spinal cord injury. Do you think it would be compatible as a material and the properties?
So you're testing my knowledge of anatomy and biology here. So I think there's I don't see any reason why our material shouldn't work in that sort of space. It's I wouldn't say it's bio-inert, but it's we've not seen any negative reaction to it in any test that we've done so far. I mean, I think you can only ever tell once you've tried something in it, first in an in vivo model, and then in vitro model, then potentially going to in vivo. But we've tested our material against I think we've done about 5 or 6 cell types they all love it. You know, there's no negative. So I've got no reason to think it wouldn't be compatible as a material. As I said, we can go from something that's as soft as a fatty tissue to something that's fairly stiff, you know, not quite as stiff as bone, but you know, up in that sort of region. So you know, we can probably have a go. So I'd say if you're interested, drop me an email. I'll connect you with our CEO.
Good. So waiting for more questions. I have a couple in the meantime. You mentioned that you realized there was a gap in the translational research within universities. I would like you to elaborate a bit more on that because I have a feeling that sometimes we like talking about translational research, but translational is more like translated to the lab next to our lab, not really translating to a different field and collaborating between chemists and pharmacologists and biochemists and so on.
Yeah, I think it's about realizing what it's going to take to make a commercial product. And I think in academia, we're often kind of insulated from that in reality because we live in our perfect little labs with our amazing equipment, and we don't focus on the things that in some products cases might make the difference between it being commercially viable and not. So in our example, some of the challenges I think we faced going from my lab, which we've got a perfectly, workable system, we could make it not a problem, and we could take it. And then you start to take it to the lab in Nottingham, and you know what? They don't have quite as nice of facilities, and the sorts of lines that we take for granted, and the atmospheres and the equipment to test what they've made, and it becomes more of a challenge that you don't realize, and you start to unwind through it, and you start to ask the questions, that you never asked before because you didn't have to, right? So what did I care about for getting that paper out that we were about to publish? Well, what I cared about for getting that paper out was how do we make enough that we can make the things that we need to make to do the cell testing, to print, et cetera, et cetera. I didn't care what the yield was. It wasn't important to me at all. I just cared that we had the material. You go to an industry lab and, and you think it's efficient, and students told you it's efficient. Well, 50% to them of a yield might be great, but in the commercial world, that's terrible. You're only using half of your thing. So you end up actually looking at things and going, actually, that's not as good as we thought it was, and that's not as good as we thought it was. That's not as easy as I thought it was, or that's, once you start to scale things up, it doesn't matter that, you can only work in 10 weight% volume of solid in solvent when you're in a lab working on a couple of 100 ml scale. When you want to get to 10 litre scale, that's a lot of solvent, and it's those little things that you don't think about as an academic because they're not important, because of the scale you're working on, because frankly, again, we kind of roll around in money really compared to a startup. And so it's not, a little bit more to buy some more because it's not as efficient. It doesn't really matter because you're not making that much of it. But when you want to commercialize something, certainly a material, you need to be able to make more of it. Therefore, the efficiency and the cost of those things becomes more important, and the processes become more important to be able to do that. And those are the sorts of gaps I'm talking about, thinking about. You don't think about them in academia, but you need to when you commercialize them. Obviously, the more you've thought about them, or the more, at least at the back of your mind as to how you could do it, the quicker that translation story will go.
We have another question from the audience. Great talk, Andrew, very interesting. Can you control the degradation rates of the material? Have you considered non-degradable materials that can be used for the development of long-term / permanent implantable devices?
So we can make materials that don't degrade. There's loads of those on the market. In terms of, you can 3D print loads of different materials that won't degrade significantly. And so we didn't think that there was an opportunity there. And I think it's about finding your USP, right? What's novel about your research? But it's not necessarily what's novel, but where's the gap in the market? So there wasn't really if you want to be a medical device company that makes medical devices that are permanent, you've got, you want to make it by 3D printing. The world's your oyster in terms of the materials you can choose. If you want to be a 3D printing medical devices company that makes degradable ones, you've now got one choice on the market effectively to go for that really does what it says on the tin. So so that's why we focused on degradable rather than non-degradable, because it's about competing as well. So there's a company called DSM in the Netherlands that make about 40% of all 3D printing resins around the world, including most of the non-degradable ones, or a lot of the non-degradable ones. And you're not going to compete with a company like that on volume, on price point, because they just have the expertise and they have the scale, and they can just scale you out in the market. So I think you need to find a real niche if you're going to spin out a company that is needed, but there's an opportunity in that other people aren't going to trample all over you on too quickly. In terms of the technical question, yes, we can control the degradation. It's rather formulation dependent, but it's a very complicated formulation. So you can kind of tune one thing down and turn something else up to retain mechanical properties and degradation. But as with most materials, it's a complicated equation of, if you want something to degrade faster, you might have to compromise your mechanical properties a little bit as well. So it's a bit of a, I say, yes, we can. It was on one of the early slides, some of the data that we can get of how quickly those materials degrade.
Unless it's almost time, I'll just finish up with one question I have. Has it been 3D printing technology something that has enhanced your project, or were you ahead of the technology and sort of have to adapt to the current state of the art for the 3D printing?
3D printing, the type of technology we're using has been around for 20, 30 years. I mean, it's just that I think, material scientists haven't really looked at it as an opportunity. Right place, right time, opportunity presented itself to us to an extent to look at that. But, with 3D printing technology, most of them are quite old. There's a few newer ones coming out that can use photocurable resins. And again, because we're really lucky that we're hooked in well with a really good 3D printing consultant, now he's finding these opportunities to look at collaborating with the companies that commercialize those types of technologies as well. So, at the end of the day, what we have is a photocurable resin, and we have a way to control the light penetration depth and reactivity of that to create good resolution. And that then can be applied in any 3D printer. I mean, I hope there's no 3D printing people in the audience, but it's not that elegant a technology, right? It's kind of neat and it's cool, but it's not that complicated. Actually, it's quite simple. We built our own. That's how simple it is, right? And so no, the benefits that the technology can offer to this sort of space are huge, in terms of not having to carry stock, in terms of personalization, or producing different things for different people per batch. It's got huge advantages in that really are useful for this space. But as a technology itself, it has its nuances, but no, there's nothing innovative required in the 3D printing to make the resins work, if you know what I mean.
Right. Okay, well, I won't take more time of you, Andrew. I should say I wrote an entire page with points that were interesting and questions. I've really enjoyed your talk, and I'm sure it's been the same for the rest of the audience. With this, I would like to thank you for your time today, for an excellent presentation and a great discussion. And I would like to let our audience know that our next Innovation Forum event would be on the 23rd of June, and it'll be on career progression in science. So make sure you check the Eventbrite page for Innovation Forum on our social networks, so you can get to know which speakers will be having that date. And without further ado, Andrew, thank you very much for coming today. And hopefully see you next time when you are going to IPO and we can accelerate it in the next webinar, Innovation Forum.
Thank you very much. Thanks for the invite.
Thank you.
Thanks. Bye.